All right, are there any questions? Otherwise, I think it's time to get started with today's lecture. So today we are going to, hopefully by the time we finish today's lecture, you will know what proteins are made of, all right? What lipids are made of, what DNA and RNA are made of, all right? What these different sugars are made of, right? So if you look at a typical cell, this is an important distinction between organic chemistry and inorganic chemistry, is that the molecules that you encounter in organic chemistry or in the chemistry of living organisms are really what are called macromolecules. They are huge in size. Okay, it's not going to be like sodium chloride. It's going to be something with a huge formula. Okay, so cells contain 4 major families of small organic molecules or modular units which are combined together to form these large macromolecules. So although you have huge molecules, all right, in living organisms you encounter huge molecules, but these are made up of smaller modular units, right? And we will focus attention on the 4 important classes of modular units. One class of molecules are what are called sugars, which are the building blocks for more complex sugars and carbohydrates, and we'll get to that in a moment. Then we'll also look at fatty acids, which are the building blocks for fats, lipids, and all cell membranes. Then we look at amino acids, which are the building blocks for proteins. And then we look at nucleotides, which are the building blocks for nucleic acids such as DNA and RNA. Right. So that's what we are going to consider in this chapter. Okay, now if you're looking at sugars, the simplest sugars, and they're called monosaccharides, these are compounds with this general formula CH2O and then N, where N is usually 3, 4, 5, 6, or 7. Okay. So because it contains a carbon, this is C and then H2O, so it's a carbohydrate. Okay. So sugars are also called carbohydrates because of this general formula, right? Now, these sugars, all right, that have this general formula with N equal to 3, 4, 5, 6, or 7, they are called monosaccharides, okay? And they usually occur as aldehydes or ketones. Recall in the last chapter we talked about aldehydes, okay, R, C double bonded with O and then H, okay? We talked about ketones, C double bonded with O, okay? We didn't do it just for the fun of it. The thing is that the sugars that you're going to encounter in biology, right, they usually occur as aldehydes or ketones. And I'll give you examples of that on the next slide and beyond. So of particular interest to us from the point of view of molecular biology are the 5 carbon sugars and the 6 carbon sugars. So here you had CH2O and then N could be any number. If N is 5, that's a 5 carbon sugar, right? Or a or what is called a pentose, all right? 5 carbon sugar. If N is 6, then that would be a 6 carbon sugar or hexose, all right? So if you look at pentoses, all right, there are again 2 different forms, all right? There could be more too, but the 2 that are of interest are this one, ribose, all right, which is an aldehyde. So see, you have the C double bonded with O and then H group here. Somebody asked me last time what was a group, an organic group. This can be another organic group, okay? Because remember, in the case of aldehydes, we had R, C double bonded with O and H, and R was an organic group, okay? So an organic group could be something as big as this, right? So here, the general formula for this one is CH2O, N equal to 5. So that's C5H10O5, okay? And this has the name ribose, and it's an aldehyde. The same one, right, the same sugar, right, you could have a different, with the same molecular formula, could have a different structural formula. And this time it takes the form of a ketone, right? Ketone, again, if you remember from the last lecture, ketone is characterized by the presence of what is called the carbonyl group, or C double bonded with O. There's some organic group in here and another organic group in here, okay? So C double bonded with O, R1 connected on one side, R2 connected on the other, right? So this is an example of a ketone, and this particular one is called ribulose, right? And you can have examples of, monosaccharides having the aldehyde and ketone forms for many different values of N, but our primary interest here is for N equal to 5 and N equal to 6, okay? N equal to 5 gives you pentoses, and pentoses are the ones that are the sugars that appear in DNA and RNA. N equal to 6 will give you glucose, and glucose is something that you know the utility of that, okay? If you're an athlete, or you know that we get most of our energy from glucose. So for N equal to 6, that's a 6 carbon sugar or a hexose. And the aldehyde form, again, C double bonded with OH, and then all of this stuff here, that is called glucose. On the other hand, if it is a ketone, C double bonded with O, then that's called fructose, right? Now, the 5-carbon sugar ribose, right, that I showed you, the aldehyde with 5 carbons, right, and its derivative deoxyribose, which I'll define in a few minutes, right? These are important constituents of nucleic acids such as DNA and RNA, right? DNA, again, somebody pointed out last time, is the abbreviation for deoxyribonucleic acid. RNA is the abbreviation for ribonucleic acid, right? And these are DNA and RNA, they contain some sugars, okay? If it is DNA, it's going to be deoxyribose. If it's RNA, it's going to be ribose. And regarding glucose, right, the 6-carbon sugar that is called glucose, it serves as an important source of energy, right, in all living organisms, right? Because we get a lot of our energy by basically from the oxidation of glucose, right? If the level of glucose in your blood falls too low, then you're in trouble, you'll pass out. All right, any questions? Next, I want to point out something to you about ring formation in these organic compounds, in these sugars, okay? So ring formation of these sugars, these 5 and 6 carbon sugars in aqueous solution. See, the inside of the cell is watery, as I said before, right? It's an aqueous solution. The boundary is made up of lipids, that's oily, right? So in aqueous solution, what happens is that the aldehyde or the ketone group of a sugar molecule tends to react with the hydroxyl group of the same molecule, thereby closing the molecule into a ring, right? And let me go back to that picture. So this is for ribose, right? Let's go back to the picture where we showed a ribose, and let's see how the ring is formed, right? So this is ribose, the 5-carbon sugar. All right, you now number the carbons like this: 1, 2, 3, 4, 5, right? Now here you have the aldehyde group, C double bonded with O and H, okay? What is going to happen is that in aqueous solution, all right, this thing here is going to come and bond here, okay? So the oxygen will connect this carbon to that 4th carbon here, okay? And the hydrogen gets removed, it's absorbed in the water, right? That connection happens, and then this guy here, the carbon number 5, along with the 2 hydrogens and the hydroxyl group, is going to stick out. So this whole thing is going to be connected, let's say in the plane of this board, right? Whereas this thing will be sticking out, okay? And that's what I'm showing in that diagram over there. So that 1, 2, 3, 4, 1, 2, 3, 4, 5, that's the labeling, okay, in that ring compound. So in aqueous solution, this will form a ring compound, right? And the same thing is true for the case of glucose, because then you have 1, 2, 3, 4, 5. There'll be a ring formation. The oxygen will come here. The hydroxyl will move onto this guy, right? And then the CH2OH is going to stick out of the plane of the board, right? That's what we are trying to show in that picture. So in aqueous solution, and in most books on biochemistry, even in medicine and all, you will see this structure. When they show ribose, it'll be this structure because it is an aqueous solution. So there's a ring compound that is formed. See here, you can see 1, 2, 3, 4. I said 1 and 4 would get connected with that oxygen, right? And then hydroxyl sticking out, and then CH2OH containing the 5 carbon, that's going to stick out of the plane of this board. So this is what ribose would look like in aqueous solution, right? Similarly, if you're looking at glucose, the 6 carbon sugar, again 1, 2, 3, 4, 5, right? The 5th carbon is connected to the 1st carbon through this oxygen, right? Hydroxyl sticking out here, and then CH2OH containing the 6th carbon is sticking out of this plane, right? So since the environment inside a cell is aqueous, it is this ring structure that you're going to be encounter over and over again, not only in this course. If you open any book on biochemistry, right, it's going to have this structure, right? It's not going to have the other aldehyde structure, right? Or even, books pertaining to medicine and all will also have this, okay? And glucose is extremely, extremely important, right? From the medical point of view. Yes, sir.
So is it possible for the reservoir group, something similar to glucose, but it has 6 carbons on the ring?
6 what?
6 carbons on the ring. I mean, so the last one is not going to stick out, but it's on the ring also. So that's the last one.
Yeah, it's possible. It's possible, but again, I have to give you because I have not looked into that. So you're saying with 7, yeah, because we said that 7 was also an allowed number for a monosaccharide. It's possible, but I just don't know whether it exists or not. Okay, it is possible, and in fact, here also there will be. See, this is glucose, right? What happens if you go and interchange the hydrogen and the hydroxyl? You'll get a different compound. Okay. In organic chemistry, if the structure is different, it will have different properties. I'll give you a couple of examples of that. Okay. So instead of glucose, it will be some other sugar. Okay. Like it could be galactose. All right. I forget which one. Either it's number 2 or number 4. If you interchange the hydrogen and hydroxyl, you get a different sugar. Right. So the possibilities are endless. But again, in this course, we are just trying to get the basics to get us to genomic signal processing. So we are not delving into that. But if you were going to med school, they would spend a lot of time. Okay. Or if you were in the biology department. All right, any other questions? So yeah, we got to your, no, actually not your question, but we'll get to that, the answer to your question, okay, is coming. Now, we have talked about ribose. Ribose is the 5-carbon sugar, 1, 2, 3, 4, 5, right? And it has this structure in aqueous solution. Now, you will notice that I have put 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime here, okay? I didn't put 1, 2, 3, 4, 5, okay? There is a reason for this, because most of the times that you're working with this ribose or deoxyribose, all right, it is going to be as part of a DNA or RNA molecule. Okay, so the sugar is only part of the mix. You know, you need some other compound, right? Some molecule plus a sugar plus a phosphate to get your DNA or RNA, right? And that first molecule, okay, it's a nitrogen-containing ring compound, that takes up the lettering 1, 2, 3, 4, 5, 6. You know, it can even go up to 9. We'll see that by the time we get done with this lecture. So that's why when you're labeling the sugar, usually you will put 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay, that's what you will see in many different textbooks, even medical books also. When they're writing the sugar, they will not write with 1, 2, 3, 4, 5. They'll write with 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Okay, so just some notation I'm letting you know. Right. But we'll encounter that later on. Now, if you start with ribose, all right, and here you have a hydroxyl group at the 2 prime location, all right? If you replace this hydroxyl group with hydrogen, that means you have taken away oxygen from this guy, okay? Then you get a new sugar which is called deoxyribose, okay? So the only difference between this and this is that instead of a hydroxyl group, you have a hydrogen. Oxygen has been taken away. This is deoxyribose. This is the sugar that is found in DNA, okay? Whether it's from you, from me, from any animal, okay? It is this sugar that is found, okay? And this is one of the things that contributes to the terminology deoxyribonucleic acid for DNA, okay, because this sugar is deoxyribose. If it is RNA is ribonucleic acid, the sugar is going to be ribose. It is not going to be deoxyribose. Important difference between DNA and RNA. And again, we are going to be encountering it again in this chapter, and later on there is an entire chapter that is devoted to DNA. So, you know, this is a theme that will run, and it will give you more opportunities to, you know, kind of master the biology, or at least pick up all the terminology and all that. Okay, but you have to keep on studying as I, you know, make progress in the lecture. You know, there's no time to relax. So this is ribose, and that's deoxyribose, right? Now, another thing that we have to understand is that in inorganic chemistry, right, quite often when you just get the chemical formula, that's the end of the story, because there is only one possible structure for that compound, right? A unique structural formula. In organic chemistry, on the other hand, compounds having the same chemical formula may have totally different structural formula and properties, right? And compounds which have the same chemical formula but different structural formula are called isomers of each other. And organic chemistry is filled with instances of isomers whose different properties make them especially well suited for particular biological functions. Like, see, even in the food that you eat, okay, you're taking in certain molecules, right? You have, you know, necessary enzymes to process these molecules. If somebody gives you exactly same molecular formula for something that has the structure, different structural formula, right? You probably die because you cannot process that, okay? That is the reason, for example, why cows, right? They can eat grass and survive, okay? If we go and eat grass, we don't have the enzymes to metabolize, you know, we'll probably die, okay? Or end up in the emergency unit or hospital or ICU, you know, whatever. And, you know, the cows and some of these other animals, you know, they will have the enzymes, the necessary chemicals that are needed to change one isomer into another, okay, where the body can process it. So a few examples of isomers, and that gets to your question. For example, if we interchange the hydrogen and the hydroxyl group at carbon number 4 in glucose, right, we obtain and you don't have to memorize all this, okay? You're not a student of biology, but this is just for your information. We obtain a different sugar which is called galactose, right? And this is the sugar that actually goes into the production of milk, right? Milk has lactose, that is galactose combined with glucose will give you galactose. And I'll show you how they link up, right? Another example, if we interchange the hydrogen and the hydroxyl groups at carbon number 2 in glucose, we obtain the sugar that is called mannose, okay? And there are lots and lots of examples no, your EE students, no memorization is required. Okay, but you have to know, be aware that these things are there. Yeah, go ahead. Isomer means it's the same molecular formula, right? But different structural formula. The formula for the compound is exactly the same. Like if it's glucose, it's C6H12O6. Okay. But then you can have many different forms. How the elements are arranged, right? How the atoms are arranged in that molecule are going to be different. Okay. Alright, so we talked about monosaccharides, or the simplest sugars that had the general formula CH2ON, where N was like 3, 4, 5, 6, and 7. Now we want to see how these units of sugar, alright, are linked together to form, let's say, disaccharides if you have two monosaccharides linked together. Trisaccharides, if 3 of them, then you can have polysaccharides also, right? So the monosaccharide building blocks can be combined together to yield more complex sugars, right? Disaccharides are made up of two monosaccharides that are linked together in a condensation reaction. Okay, condensation reactions are reactions where two units are linked together and a water molecule is eliminated. So say for example you have some sugar here, another sugar here, monosaccharides, and you know that at all these different locations you have hydrogen and hydroxyl groups sticking on, right? So between these two guys you can eliminate a water molecule, right, H2O, and you will have an oxygen linking these two guys together, right? Now you got a disaccharide that has got two sugars that are linked together, okay? Guess what? This can be continued because there are hydrogen and hydroxyl groups elsewhere, okay? You can link up more. Even not only that, it doesn't even have to be a straight chain. It could be a branch chain. You might have a linkage here, then another one here, and you can have all kinds of weird shapes, right? I mean, that's the reason why organic chemistry is so difficult, right? Because there are so many possibilities, okay? So since there are many hydroxyl groups on each monosaccharide, two monosaccharides can link together in many different ways, and due to the availability of additional hydroxyl groups on the disaccharide, more monosaccharides can get linked to it, producing chains and branches of various lengths. If these are short chains, they are called oligosaccharides, while long chains are called polysaccharides. Right? An example of a polysaccharide is glycogen, right, which is made up entirely of glucose units that are linked together. Right? In fact, glycogen serves as the body's energy reserve, because you need to have your blood, the level of glucose in your blood. Okay, you need to have it within limits, because red blood cells can only use glucose. Right? Your brain needs glucose. Right? If your glucose drops, your brain doesn't get glucose. Right? For 4 minutes. Right? Well, oxygen and glucose, it doesn't get energy. You're brain dead. Okay. Yeah, and we don't know how to revive somebody that is brain dead. Your organs function, but you're basically gone. So it's that important. Right? Now glycogen serves, and the body has mechanisms for regulating the level of glucose, right, in the blood. It has to be within certain tight limits. But let's say the level of glucose in your blood goes up. Okay. It's too high, then the excess glucose needs to be stored somewhere. It can be stored as glycogen, as units, glucose units that are linked together, right? And we all have about 1 day's reserve of energy, right, stored in the form of glycogen in the body, right, for about a day. But it's not very because glucose has got water in it, right? So if it's stored as glycogen, it's not very compact storage. So first, if you're eating too much, okay, if you're eating more than you're exercising, so your blood glucose is high. So the excess glucose will first get stored as glycogen, but that's only a day's supply of energy. Beyond that, it'll start getting stored in the form of fat, okay? Because no matter how much we dislike fat, right, it is a very efficient storage of energy, because it can store, the same weight of fat will have like about 6 times as much energy as glucose, right? So that's an efficient storage mechanism. So after that, it'll store it as fat in the body, okay? So glycogen is an example of a polysaccharide. This cellulose is an example of a polysaccharide, okay, produced by a plant, right? This wood, the polysaccharide. Now, the kind of reaction that I showed you, where two units are linked together by the elimination of a water molecule, that's called a condensation reaction. But these reactions are not unique to the formation of sugars from monosaccharides. In fact, they are used to form proteins from amino acids and nucleic acids from nucleotides. In this chapter, I'm going to talk about amino acids and nucleotides, and I will show you how amino acids can be linked together to produce proteins. Right? Nucleotides can be linked together to produce nucleic acids. Right? Now, so If you have smaller units, you're trying to link them together, produce a larger unit, that's called a condensation reaction, removal of a water molecule. The reverse reaction of condensation is called hydrolysis. Hydro is water and lysis means to split, right? So hydrolysis is splitting with water, right? And this plays an important role in our digestion of carbohydrates and other foods, right? Because if you eat like complex carbohydrates, that's what you're supposed to eat, right? Maybe not for you guys are young, but for older people, they'll tell you to eat complex carbohydrates because, that's better for your body. You do not want to have wild swings in your blood glucose, right? It should stay around some, reasonable value, right? So, if you have these complex carbohydrates, naturally that's made up of many of these monosaccharides that are linked together. The body can only absorb glucose, right? So these have to be broken down, okay? So there have to be hydrolysis reactions, right, that break those down into the simpler molecules that the body can absorb, right? And that is, carried out by different enzymes. For example, your saliva has got enzymes that can break down glucose, right? Then even in your pancreas and other places, you have enzymes that will break down these complex molecules into, simpler molecules that the body can absorb. Because in biology, there's no shortcut, okay? You cannot just eat some meat and immediately put on some muscle or something like that. That has to be broken up into at the molecular level. Then it has to go through the system, then be absorbed, and then you build up muscle. So there are no shortcuts there. Now, some specific examples of disaccharides resulting from linking together two monosaccharide units, and this list is by no means exhaustive. Okay, that's not my goal here, but just to give you an idea. For example, two glucose units can link together to produce maltose. And again, the linkage will have to be at the right location. Okay, I don't know what the location is, whether it's 3', 4', 5, I don't know. But if you open a book on biochemistry, they'll have that information, right? Or these days, you don't even need to open a book. You just do Google search or go to Wikipedia or whatever, and you'll get that information. Glucose and galactose, when they link together, that produces lactose, which is the sugar found in milk, right? That means if you drink milk, right, you have lactose. To break down that, so you'll have to break down that lactose into glucose and galactose, right? For that, you need enzymes, right? And like we have that enzyme that's called lactase, right? And a lot of, I think, people from Asia or something like that, if they're older, they develop lactose intolerance, right? Because you stop producing lactase, right? And then try drinking a glass of milk if you're, lactose intolerant. You see how much fun it is. So. Similarly, glucose and fruit. So that tells you, even if you have isomers or you have the compounds, if you don't have the right enzyme, then you're in trouble. Glucose plus fructose is sucrose, right? It'll give you sucrose if you link the two of them together. Now, one thing I should point out, I think somebody brought this up in the last lecture also about the energy considerations and reactions. These reactions are not energetically favorable because they're generating order, right? Glucose plus glucose, you're not breaking down something, you're building up, okay? So for these reactions to take place, you need certain extra factors to kick in. These will not happen spontaneously, right? So just keep that in mind, and we will see that again in the next chapter. All right, so we finished our discussion of sugars. All right, they're made up of those monosaccharides, and we have seen how they get linked up together. The next class of modular units that we are going to look at are what are called fatty acids. Right? These are organic acids, right? Because we're looking at molecular biology, so these will be carboxylic acids or organic acids. So a fatty acid molecule has got two distinct regions. It'll have a long hydrocarbon tail. Okay, like here, this is carbon, 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 and hydrogens attached all over, right? So this is a hydrocarbon. We know that hydrocarbon is hydrophobic, right? I mean, because carbon and hydrogen, they attract electrons in a covalent bond to the same degree. So there's no charge distribution. So this thing will hate water. It will not interact with water, right? But on the other hand, the fatty acid will also have a carboxyl group, all right, on the other hand, C double bonded with OH. And this is a hydrophilic group, right? Because this likes water. This portion can dissociate in water, right? Or partially dissociate to produce hydrogen ions, right? So it'll have two things, a long hydrocarbon chain, and then a carboxyl group which behaves as a carboxylic acid, is ionized in solution. That means it gives up its hydrogen ion, and is extremely hydrophilic. It really loves water. So this guy loves water, this one hates water, okay? So that's a fatty acid, and you can have different examples of that. This particular one is palmitic acid, so there will be 16 carbons here, 1, 2, like that. So you count, there should be 16 carbons in here, so C16, And this is what is called a saturated fatty acid. You may have heard of that, right? Like people talk about you should have polyunsaturated fatty acids, right? Again, I'm not of interest to you guys. You having everything, whatever you eat, it works out fine, okay? But for older people, to avoid health problems, they're supposed to have unsaturated fatty acids. So what do you mean by saturated fatty acid? Means that all these carbons are linked together only by single bonds, okay? So there's no room for putting in more atoms, right? Because everything is linked by single bonds. So this is palmitic acid. This is the kind of acid that fatty acid that you see in, let's say, coconut oil, right? That's why it's palmitic acid, or a palm, or a date palm. So palmitic acid with 16 carbons in it is a saturated fatty acid. And saturated fatty acids are characterized by the absence of double bonds between the carbon atoms, right? So next, and there are lots of examples of these. I don't even know, or I didn't even care to know the formulas for all these different, fatty acids. You don't need to either, okay? But at least know that what a saturated fatty acid is. And then I give you an example of an unsaturated fatty acid. Is oleic acid. It has got 18 carbons, right? And it is unsaturated because you have a pair of carbon atoms. These are all carbon atoms. Carbon here, carbon here, carbon here, carbon here. A pair of carbon atoms that are linked together by a double bond. Okay, this is oleic acid. So guess in which fruit or vegetable or whatever are you going to find oleic acid? Can you guess? Olive oil, yeah. Yeah, olive oil is going to have that, yeah. So, but this has a double bond. So this is an unsaturated fatty acid, okay? And this double bond, remember I said that last time, okay? I think it was last time only, that creates steric hindrance, right? Because that is not going to allow you to fold things up properly. So that's why this molecule will take a particular shape, right? It's possible that that's what gives it its beneficial effect, So looking at this, you can say that probably this olive oil would be a better thing to consume than going and having palmitic acid, because that one is a saturated acid. This is at least unsaturated, right? So maybe this allows something else in the body to process it differently. I don't know. I'm just, making a guess here. All right. Any questions? Now, how are fatty acids, stored in the body? Okay, because usually, as I said, if you have excess of energy, that's going to get stored as fat, right, in the body. Right. Now, fatty acids serve as concentrated food reserves in cells, as they can be broken down to produce about 6 times as much usable energy weight for weight as glucose. Okay, so this is really compact storage, right? I mean, so the excess fat that people accumulate, right, is not that it's nature's purpose to make them look bad or whatever, or make them not feel good. It's because it is compact. Okay. Otherwise, your volume would have to be 6 times more. Okay? You'd have to be 6 times bigger to carry the same amount of energy, right? So fatty acids are usually stored in cells as triacylglycerol molecules, or what are called triglycerides, which consists of 3 fatty acids joined to a glycerol backbone. So this is a glycerol. It's an alcohol, right? See, like OH, OH, OH. This is glycerol. If you're looking at a triglyceride or triacylglycerol, what is going to happen is that this hydroxyl group over here is going to link up with a fatty acid. Remember, a fatty acid had the long hydrocarbon chain, then there was a carboxyl group. Okay, so between this OH and this OH of the carboxyl group, you can eliminate a water molecule, all right? And you can link this up with this first branch of this glycerol molecule. Then the second one will have another fatty acid, right? So different groups. R1 could be, this could be, palmitic acid, this could be oleic acid, whatever, okay? This could be yet another fatty acid. And if you have a glycerol molecule that is linked up to 3 fatty acids, that is called a triacylglycerol, or the doctors refer to it as triglycerides, because if you go and do blood work, they will check your triglycerides level, because that tells them how much of fat is swimming around in your blood, okay? Because that has the potential to, go and clog your arteries and things like that. Again, at your age, you don't have to worry about anything, so. But as you get older, those become significant factors. Okay, any questions? So this is the storage of fats as triglycerides. Next, I also want to talk about what are called phospholipids and glycolipids, all right? So again, the starting point is the glycerol molecule, right? If you're looking at a phospholipid, you will have a fatty acid that will get attached to the first location, fatty acid to the second location, And it's a phospholipid, so there'll be some phosphate group along with some hydrophilic group, something that loves water attached, okay? Now, what is the net result? The net result is that this looks like a molecule that has got two parts, because there's a fatty acid tail here, which is a hydrocarbon tail, okay? So it hates water. This one hates water, okay? This guy loves water, all right? Remember, I showed on the first day, I showed that picture, all right, that there were two portions. There was a molecule, all right, that had a head that really loved water, and then there were the two tails that hated water, all right? I showed you that. That's basically this, okay? I didn't come up with those numbers 2 and 1 just for the fun of it, you know. This is how it occurs in nature, okay? So if it's a phospholipid, this third one is going to be a phosphate attached to some hydrophilic group. If it is a glycolipid, then it's going to be some sugar which really likes water, right? That'll get attached here, right? So phospholipids are the major constituents of cell membranes. In phospholipids, 2 of the hydroxyl groups in glycerol are linked to fatty acids, while the third hydroxyl group is linked to phosphoric acid, right? And this is a schematic diagram for a typical phospholipid. And you can, and this is something that we already discussed, right? How a fatty acid can be used to form a surface film and a micelle, right? So since a fatty acid possesses a hydrophilic part and a hydrophobic part, it's an amphipathic molecule, it can form a surface film or small micelles, okay? So if you put water and you just scatter some fatty acid, the part that loves water will stay in contact with the water, the part that hates water will stay away from it, you will have a film like this, right? This is what happens when you try to mix up oil and water, right? The oil is on top and it's staying like this. Similarly, you can form what are called micelles, okay, where, you know, there is this, this is like a bubble, okay, where the exterior is hydrophilic, all right, and all the portion that hates water is staying away from it, okay. And this is actually like if you talk about, like how because see, we need to, see, our blood is actually aqueous solution, okay, it's watery, all right, but then fats have to be moved around, right? So usually fats are encapsulated in this form, all right, into something that is called a chylomicron, all right, and then it can move around because now the outside is watery, okay, you have fat inside and it'll go to the relevant tissue, maybe you need to build up some fat over there or some, you know, fat is needed for cell membranes and so on, okay, so that's how the transportation takes place, right? So another example is the formation of the lipid bilayer, which I discussed in the first chapter. So because of similar reasons, the phospholipids and glycolipids, which are lipids made up of 2 fatty acids for the hydrophobic part and 1 or more sugar residues for the hydrophilic part, they form these self-sealing lipid bilayers, which are the basis for all cellular membranes. So again, the 2 hydrophobic tails, a hydrophilic head, in an aqueous environment, right? This will form, this whole thing will become like a bilayer where the hydrophilic part is facing the water, all right? Hydrophobic part is staying away from the water. If you go and try to mess it up, you stick a needle in, right? It'll create an opening, but when you take the needle out again, it's a self-sealing membrane, right? That's pretty nice, right? How many things seal by themselves? Not too many. Right? If you break something, it doesn't fix itself automatically. Here it does have the ability to do that. All right, any questions? So we already covered sugars, all right? We also covered fatty acids, all right? So next we are going to start looking at amino acids, which are the building blocks of proteins. So before I start on that, any questions on whatever we have covered so far? Now, amino acids are the subunits of proteins. They possess both, it's called amino because it's an amino acid. So there will be an amino group somewhere, there will be a carboxyl group somewhere, okay? That's why it's an amino acid. So amino acids are the subunits of proteins. They possess both a carboxylic acid group and an amino group, both linked to a single carbon atom called the alpha carbon. So here you have this single carbon atom that's called the alpha carbon. On one end you have this amino group, NH2 is the amino group. On the other end you have the carboxyl group, C double bonded with O, H, alright? Carboxyl group. And then you have a hydrogen atom, and then you have something here that is called the side chain group, right? So all the amino acids will have this structure. The difference between the different amino acids will come in the choice of this side chain group, right? This R will be different for different amino acids, okay? And you know, you're all engineers. You know, if I have amino acids linked together, okay, let's say 20 amino acids, right? In each location, I have possibilities for 20 different amino acids, okay? So if I have a chain that is 100 units long, all right? So the number of different compounds that you can come up with is like 20 to the power of 100, okay? Really large number, right? Many, many possibilities. Right? So amino acids are the subunits of proteins. They possess both a carboxylic acid group and an amino group, both linked to a single carbon atom called the alpha carbon. Amino acids differ from each other because of the different side chains, these guys, R's, that are also attached to the alpha carbon. And a typical amino acid has the following structural formula, right? Now, if you look at any living organism, right, it will have amino acids, right, with 20 different possible side chains. Okay, for all life on this planet, we'll have amino acids where R will be one of those 20 possibilities. Okay, if you have life on some other planet and it is amino acid based, you might have additional side chains. Okay, but that's not been discovered so far. Okay. And these things actually point to a common ancestor too. Okay? I mean, like if even from the simplest bacterium to humans, okay, if you have the same amino acids, okay, but it's the different combinations that will make the functions different, okay, of the different proteins. Okay, now I give you a listing of the different possibilities for the side chains. Again, you don't have to memorize anything, but if you open a book on organic chemistry, molecular biology, they will give you the detailed structure. What those R's are, the individual R groups, they will put out the detailed structures. Okay, I'll just summarize that. So first, you have basic side chains. Okay, that means the side chain that you're going to have, that R is going to have basic properties. So these side chains usually contain a nitrogen atom or an amino group. We know that amino group is a basic group because it reduces the hydrogen ion concentration in solution. Okay, it'll take up a hydrogen ion to form the ammonium ion. Okay, we know that. Okay, I already discussed that before. So these are basic side chains. So examples of amino acids with basic side chains are lysine, arginine, and histidine. And for us right now, all that is Greek. Okay, because if you open a book on molecular biology, or even for people books that people that go to medical school use, they will give you the detailed structure. Okay, if you're a molecular biology student, you would need to know what the detailed structure is. Right. Then you can have acidic side chains where in the side chain you will have another carboxyl group. Right. So examples of acidic side chains are if R is CH2COOH. So you can see this is a carboxylic acid, right? Then you have what is called aspartic acid, right? It's an amino acid, but it's called aspartic acid or aspartate. Another example is if you have R equal to C2H4COOH, then this particular amino acid is called glutamic acid or glutamate, right? Then yet another possibility would be to have uncharged polar side chains, right? So these amino acids contain a hydrophilic side chain containing an amino group or a hydroxyl group. And examples of amino acids with uncharged polar side chains are asparagine, glutamine, serine, threonine, and tyrosine, right? And you can count. If you count all these names, you'll see there are 20 of them, like 3 here, then 2 here, right? Then another 1, 2, 3, 4, 5 here, right? And then count the rest, you'll see there are only 20 different side chains, okay. Then the last one, these are non-polar side chains. These side chains contain hydrocarbons, right? Non-polar, these are like hydrocarbons. Carbon and hydrogen, remember again, carbon and hydrogen attract electrons to the same degree. So carbon-hydrogen covalent bond is non-polar, right? It's not going to mix with water. So these side chains contain hydrocarbons and are usually hydrophobic or non-polar. And examples of amino acids with non-polar side chains are alanine, valine, leucine, isoleucine, proline, phenylalanine, methionine, tryptophan, glycine, and cysteine. And there are 20 different amino acids. I'm just going to list the structure for just one of them on the next slide. Okay? If you're interested in more structures, you can go and look at the books. Even if you type this, maybe even on the internet, it will show you the structure. So this is a representative amino acid. This is called cysteine, right? So this is the amino group. This is the carboxyl group. This is the hydrogen connected to the alpha carbon, and then this is the side chain. Notice that this side chain has got sulfur in it, okay? So in fact, if you have 2 of these guys, they can link together, okay? The side chains can link together because the 2 sulfur elements can link together and the hydrogen can be eliminated, okay? Inside the cell, that will not going to happen because inside the cell, it's an aqueous environment. So you have a lot of hydrogen atoms or hydrogen ions moving around, right? But when it's outside the cell, you could have units link up together, cysteine units link up together like this. So disulfide bonds can form between 2 cysteine side chains and can play an important role in determining the 3-dimensional shape of the protein containing these cysteines. Okay? So if you have something that has cysteine inside the cell and it's exported from the cell, outside the cell there isn't that much of hydrogen, so you might have disulfide bonds that might actually cement the shape of that protein. Right? But again, this is something that we are going to encounter later on in the chapter on proteins. Right? Now in this chapter, the important thing to note is that you have this amino acid with an amino group at one end and a carboxyl group at the other. You can take two different amino acids, okay? Link the amino group of one with the carboxyl group of the next one, right? Through a condensation reaction, and you have produced, two amino acids that are linked together, right? You can continue the process, right? And that's how you build up proteins. So amino acids can be linked to each other via peptide bonds, which are essentially amide linkages. Again, that's the reason why I introduced those amide linkages in the last lecture, okay? When I talked about, alcohols and carboxylic acid reacting together to produce an ester, okay? One example of an ester is an amide linkage. So you can look at this picture here. You have amino acid number 1 with side group R1 or side chain R1, amino acid number 2 with side chain R2. You have the amino group of this guy and the carboxyl group of this guy. Between the two of them, a water molecule is eliminated. And then you have this C double bonded with O is linked up with NH, and you have this peptide bond. Okay. You can continue the process, and this is a condensation reaction because the reaction was achieved through the elimination of a water molecule, right? Now, even after you've done the first linkage, now again I still have an amino group over here, right? A carboxyl group here. So I can do it again. I can link up the amino group of the next amino acid with this carboxyl unit and continue the chain, okay? And it can be very long, right? So if you're looking at a protein, which is basically a bunch of amino acids that are linked together by peptide bonds, on one end you will have what is called the amino terminus or the N terminus, right, of the protein. So it has a very definite polarity, and on the other end you're going to have the carboxyl terminus or the C terminus of the protein. So here when we are linking up amino acids, we have a well-defined directionality, an amino group at one end and a carboxyl group on the other. It is possible to attach additional amino acids to the existing chain via further condensation reactions. And the final chain that you have will also have an amino group at one end, that's called the amino terminus or the N terminus, and a carboxyl group at the other called the carboxyl terminus or the C terminus. Right, and this is the same picture that is repeated here. Amino terminus on one side, carboxyl terminus on the other, and then there is this peptide bond. Okay, and these two guys, C and C, are the two alpha carbons coming from the two amino acids that we have linked together. Now the four atoms inside this block, right, the C, N, O, and H, they form a rigid planar unit so that there is no rotation about the carbon-nitrogen bond. Okay, that's just the way it is. You cannot rotate this carbon and nitrogen relative to each other. However, the two bonds that are linking the alpha carbons, this C here to this one and this one, they allow rotation. So the long chains of amino acids are very, very flexible, right? You can rotate it. And being engineering students, that, things always move towards the conformation of lowest energy, right? So this will fold up. If you have a bunch of different amino acids with different side chains, this thing will fold up based on many different considerations, okay? The effect of all the different factors will be summed together, right, in some nonlinear fashion. That will give you your final shape for the protein, okay? And that is what is going to determine the function of the protein, whether it functions properly or not. So since polypeptides can be of any length, and each location we can have 1 out of 20 possible amino acids, 20 possible side chains, there is an enormous variety of proteins that can be synthesized using peptide bonds between amino acids, okay? But biologically, the proteins, the useful proteins are the ones that have a fixed three-dimensional conformation, okay? You do not want something that falls, today in some shape and tomorrow in some other shape, okay? That's not, like if you're looking at hemoglobin in the blood, that has got exactly the right shape, okay? Exactly the right shape for binding oxygen, for the job that it is designed to do, picking up oxygen from the lungs, taking it to the tissues for cellular respiration. It has exactly that right shape. In fact, hemoglobin is made up of two units. There's an alpha unit and a beta subunit, right? The beta subunit is made up of 146 different amino acids. If you go and change one of those amino acids, okay, just one, right, that's enough to mess up the hemoglobin completely, right? And if your hemoglobin is completely messed up, you're dead, basically. So there are many different possibilities, but only the ones which we see in nature, those are the proteins that have got unique shape, right? That means they will fold into a particular shape that is needed for their function, right? If you change the shape a little bit, you know what happens. For example, you know, we are all alive because we have functional hemoglobin, okay? Let's say one of us doesn't have functional hemoglobin. What is going to happen? Okay, you're going to die. You're going to get eliminated from the population. So what we are seeing right now is basically what works, right, in living organisms, right? Okay, and there will be a detailed chapter, all right, on these proteins, all right. There's, in fact, it's probably one of the longest chapters in the book, so we will encounter this again, you know. So in this chapter, we are just trying to lay the key concepts, all right, and the building blocks, and try to gain some understanding, and more detailed treatment is coming. So any questions so far? So we talked about sugars, all right. Then we talked about amino acids, we talked about fatty acids, all right. The last set of modular building blocks that we want to look at are what are called nucleotides. These are the subunits of nucleic acids such as DNA and RNA. A nucleotide is a molecule which is made up of a nitrogen-containing compound which is linked to a 5-carbon sugar. That's a pentose, right? It could be either ribose or deoxyribose, and this is going to carry one or more phosphate groups. The nitrogen-containing ring compounds, these are called the bases of DNA and RNA, or of nucleic acids. They are of two types. They're pyrimidines and purines, right? So there are two types. If it has a 6-member ring labeled like this, remember I told you earlier that, you know, the numbers without primes are taken up by these guys, right? So the sugar will have primes on it, the carbon in the sugar. So here, this is a pyrimidine, a 6-member ring, or you could have a 9-member ring that would be called a purine, right? Now so next I give you examples of pyrimidines and purines, right? So if you're looking at pyrimidines, there are 3 of them. One of them is called cytosine, that has this structure. Again, this is a 6-member ring, 6-member ring, 6-member ring. This is cytosine, C, okay? Uracil, U, denoted by U. Thymine, denoted by T. You don't have to memorize all of this. If you want to, you know, you can, right? That's up to you, right? But, I mean, yeah, I think it would be difficult. I would like to point out that, for example, thymine, all right, it is basically 5-methyl uracil. If you look at uracil here, at location number 5, you put a methyl group CH3, right? Then you got thymine. So there is a relationship between them. You know. Again, you don't need to delve deep into this, except know that pyrimidines are these 6-member ring compounds. Okay, you have cytosine, uracil, and thymine. C and T, okay, they occur in DNA. U occurs in RNA. Okay, so these are pyrimidines. Then, in addition to that, you have things that are called purines, where you have a 9-member ring, right? So A is a purine. This is adenine. That's called a purine. Guanine is also a purine, right? And again, if you want to remember this, I took this class. I mean, I was sitting in on a class at A&M, okay, where they were teaching about this, and some students said they remember this by saying ags are pure. You know, if that helps you remember that adenine and guanine are purine, you know, feel free to use that. You know, you may believe or you may not believe it. Ags are pure, apparently. So. But again, I mean, if it is DNA, you will have A, G, C, T. Okay? If it is RNA, you will have A, G, C is common between DNA and RNA, but instead of T, you will have uracil, or U. Right? So the first component of DNA, RNA, all right, or nucleic acid, would be those bases, A, G, C, T, U. Okay? The next component is this 5-carbon sugar. If it is ribose, if it is RNA, then the sugar is going to be ribose. Okay, whose structure we just studied, 1', 2', 3', 4', 5'. This is ribose. Or you take out the oxygen from the second location, you get deoxyribose. Okay, so this is the sugar that will be present in DNA. This one is in RNA. Okay, there is one more thing missing. So it's a base, sugar, and then there is a phosphate group. Right? There's a reason why we put in that phosphate, and we talked about phosphoanhydride bonds and things like that. Okay, not just for the fun of it. You know. I mean, because this is an entire field. I wouldn't be pulling things out and putting it here, you know, because there's an entire field over there, you know. Why should I just put things here for the fun of it? You know? I have to be selective. Only whatever is needed here is covered in this class. So the third component is the inorganic phosphate ion. So here, for example, if you have C, okay, it could be anything. It could be A, G, C, T, U, okay, whatever you like. C here, cytosine. Then you have this 5-carbon sugar. In this case, we are using ribose. So let's say it is an RNA molecule that we are trying to construct, ribose. And then there is a phosphate. So how is the linkage done, right? Well, the 1' here is going to link up to this one location on the base. See? That 1' here is linked up to that N. Okay, you take this sugar, stick it in here, right? Okay. I mean, whatever, water molecule is eliminated, whatever is done, but this now is linked to that nitrogen at the bottom, right? Then you have the 5' end of the sugar sticking out, right? Attach, this is CH2OH, again, attach this inorganic phosphate here. Because there's a hydroxyl, CH2OH, you can eliminate a water molecule, you can form this phosphoester bond, right? Okay, so this gives you one unit, right? It's called a nucleoside. This is the unit from which DNA or RNA is built, right? This is one unit. So how are you going to build up a sequence? Okay, you basically connect these units end to end. And where are the ends going to be? One is this 5' end, right? The other end. So this has a polarity. There's a 5' end with this phosphate. There's a 3' end with the hydroxyl group, right? So the way you link it up is, this is one unit. Let's say it has a C. Another one might have a G, okay? Another one might have a T. Way you link it up is that this phosphate group from the next unit will come and link up over here, right? This already has one ester bond, phosphoester bond, okay? When this unit from the phosphate unit from the next block comes and links up here, you will have two ester bonds, okay? So it will become a phosphodiester bond, right? And that's how it will get linked together. So, and again, this chain can be continued because I have a phosphate on one end at the 5', the other end I have a hydroxyl group with 3' sticking out. Okay, and you can keep on linking. And then this thing will generate the sequence, the C, G, T, whatever, that is the DNA sequence. The rest of it is the sugar phosphate backbone. And again, if you didn't get the complete picture right now, you know, just be patient. We have an entire chapter that is devoted to DNA. So I will talk about this in more detail, but I think I did try to explain clearly how the linkage will take place. This is one end, okay? This is the other, right? So the phosphate of the next molecule will come and link up here. So again, now you have two units linked together, phosphate on one end, hydroxyl on the other. Again, you can link up more units, and that's how you can, and if you look at the human genome, if you look at any of our cells, there are 3 billion of these guys that are linked together. That is producing DNA, right? And the difference between DNA and RNA is if it is DNA, this will be AGCT, right? If it is RNA, it will be AGCU, right? If it is DNA, this sugar is going to be deoxyribose, means this oxygen will not be there, all right? Or if it is RNA, it will be just ribose, which is basically what you have right here. So nucleic acids are formed by stringing nucleotide units together. If the sugar is deoxyribose and the bases are AGCT, we have deoxyribonucleic acid or DNA. Sugar is ribose and the bases are AGCU, then we have ribonucleic acid or RNA. And as I said, this topic will be treated in detail in a later chapter, right? And I just want to, state a few facts and then we, dismiss for today. So in addition to serving as the building blocks for DNA and RNA, nucleotides have many other functions. They can serve as storehouses of chemical energy, and I will elaborate on that in a later chapter. They can combine with other groups to form activated carrier molecules or coenzymes. Again, this is going to be elaborated in the next chapter, how that can be done. Like they are carriers of energy, okay? It's like, if you have a battery, you can move energy from one place to the other. These guys can do that. And they are also used as specific signaling molecules in the cell. Example, cyclic AMP. Again, this will be elaborated in a later chapter, right? CAMP, right? And the details will be discussed later in this course. I believe I have almost finished this chapter just with this recap. So macromolecules found in cells contain a specific sequence of units, sugars, amino acids, and nucleotides. These macromolecules are formed by condensation reactions, where a new unit is added to a growing chain by the expulsion of a water molecule. I gave you examples of that, okay? Non-covalent bonds such as hydrogen bonds, hydrophobic forces make macromolecules assume specific shapes. I didn't discuss that in detail, but I kind of gave you the idea that many things are going to interact together, right, to form the final three-dimensional shape. And we will encounter this in the chapter on proteins. And these specific shapes, as well as the non-covalent bonds, allow macromolecules to seek out their appropriate partners and undergo the required reactions with them. So what I'm going to do is these last two slides, I will revisit them on Monday, right, before moving on to the next chapter. Okay, how about that? And if you have any questions, let me know, right? So the next chapter will get to one of your favorite topics about energy considerations, which reactions are feasible and so on.